Hi everyone. Today I am going to discuss about media formulation. This topic is coming under the paper Industrial Microbiology. An ideal production medium have some properties. The medium must have suitable chemical composition. That is, the medium should contain source of carbon, source of nitrogen, growth factors and mineral salt. Raw material should be freely available. There are two types of media, non synthetic media and synthetic media. Non synthetic media is also called crude media. Crude media have wide application. But in the case of synthetic media, the application is limited. It requires high cost. Precursors are added to the media for the better yield of the product. The medium should be free from contamination. And also, the recovery process must be easy and cheaper. The maintenance of the pH is necessary during the fermentation process. Acidic or basic compounds are accumulated in the media. To control the pH, buffers are added to the medium. Foam causes serious problem. It causes the contamination problem. To control the foam problem, we can use anti-foam agents in the media. An ideal production medium should be free from any toxic material. That is, it doesn't causes any toxicity or toxic effect on microbial growth or product formation. Let's move on to raw materials used or the materials used for the media. It includes saccharide material, starchy material, cellulosic material, hydrocarbons and vegetable oil, and finally nitrogenous material. Saccharide material include molasses, fruit juices, cheese whey, etc. Molasses is a byproduct of the sugarcane industry. 95% of the sugar in molasses are fermentable. It is rich in pantothenic acid, thiamine, phosphorus, and sulfur. Certain vitamins are also present in the molasses, that are biotin, pantothenic acid, thiamine, pyridoxine, etc. Another example for the saccharide material is the fruit juices. Fruit juices consist of sugars, soluble sugars. Example, grape juice. Then cheese whey is also an saccharide material. The straw colored liquid produced as a byproduct of cheese making is called cheese whey. Okay. So it is a waste product of the cheese or cheese industry. Second raw material is starchy material. Example, cereals, roots and tubers, etc. Cereals include wheat, rice, maize, etc. Then root and tubers include potato and tapioca. Then another raw material is the cellulosic material. Cellulosic material consists of cellulose, that is beta glucose held together with beta glucose by beta 1,4 linkage. Example for the cellulosic material is the sulfite waste liquor. It is a waste product in the paper pulp industry. Here, wood is subjected to hydrolysis by the help of calcium bisulfate under heat and pressure. This is called digestion process. At the end of this process, the spent liquid is left and it is referred to as sulfite waste liquor. So, the sulfite waste liquor is a waste product in the paper pulp industry. It contains hexoses and pentoses. Hexose means 6 carbon sugar. Okay. Example glucose, galactose, mannose, etc. And also it contains pentoses, 5 carbon sugar. Example xylose and arabinose. Then next raw material is the hydrocarbons and vegetable oils. Hydrocarbons used as fermentation substrate. Okay. Example paraffin, gas oil. These two are used for the production of SAP. Vegetable oils obtained by de-oiling of the vegetable seeds. Okay. Oils are classified into 3 types: oleic, then linoleic, and linolenic. Okay. Then last one, nitrogenous material. It include corn steep liquor, soybean meal, and pharma media. Corn steep liquor used in penicillin production. Then soybean meal is the material left after de-oiling of soybean seeds. It contains 8% nitrogen. Pharma media is a clean yellow ground powder and it is prepared from embryo of cotton seed. Let's enter into our topic, media formulation. Media formulation is essential in the manufacturing process. The medium must satisfy all the essential requirement for the cell biomass and its production. Okay. So there must be an adequate supply of the energy for biosynthesis. Carbon, nitrogen, oxygen and other nutrients gives biomass and product. Media consist of water, carbon, nitrogen source, growth factors, precursors, minerals, buffers, aeration, antifoam agents, inhibitors, precursors, and inducers. First one, water. All fermentation processes require water, but solid state fermentation require only little amount of water. Okay. It is a major component of the media and it is important for heating, cooling, cleaning, rinsing, etc. So, a large source of water is required for the fermentation process. Okay. Then, important factor to consider when accessing suitability of the water supply are its pH, dissolved salt, and effluent contamination. Then, second one, carbon source. It is required for all biosynthesis. Okay, that is, carbon source is required for the product formation and cell maintenance. The carbon source act as a source of energy. The requirement of the carbon can be determined from biomass yield coefficient. This coefficient can be denoted by Y. Okay. Factors influencing the carbon source are cost of the product, ratio at which it is metabolized, then cellular yield. Carbon sources include starch, glucose, fructose, sucrose, molasses, either cane molasses or beet molasses, then malt, etc. Third one is oils and fats. This can be used as a antifoam agent and also carbon source. Example include soya oil, olive oil, etc. Next, we can discuss nitrogen source. Industrial microbes can utilize both inorganic nitrogen and organic nitrogen. Inorganic nitrogen may be supplied as ammonium salt, such as ammonium sulfate, diammonium hydrogen phosphate, or ammonia. This can be used to adjust the pH of the fermentation. Then, organic nitrogen source include amino acids, proteins, and urea. Then, next parameter is the precursors. These are the substances added to or simultaneously with the fermentation. Okay. It doesn't causes any change in the molecule of the fermentation product. But the precursors increases the yield or the production capacity. And also it improves the quality of the product. These are required in certain fermentation process. Example, corn steep liquor or direct addition of more pure compounds. Then let's move on to growth factors. Some microorganisms cannot synthesize all cell components, so it requires certain compounds. That compounds are called growth factors. Example, vitamins, amino acids, fatty acids, or steroids. Complex media may contain most of these compounds. Next one is minerals. All microorganisms require certain mineral elements for the growth and metabolism. The essential minerals include magnesium, phosphorus, potassium, sulfur, calcium, and chlorine. Then cobalt, copper, manganese, iron, molybdenum, and zinc are also essential but in trace amount. Mineral requirement depend upon the product. That is, sulfur is used in the penicillin production, sulfur is used in the cephalosporin production, etc. Then buffers. Buffers are used to maintain the pH of the medium. As the microbial growth is affected by the pH changes, optimum pH for most organisms is 7. Commonly used buffers are calcium carbonate, sodium hydroxide, and ammonia. The balanced use of carbon to nitrogen ratio, that is CN ratio, controls the pH. pH can be controlled by adding sulfuric acid, sodium hydroxide, or ammonia. Next one is aeration. Air is required for the aeration and it is supplied to the fermenter by pumps or compressor. It is sterilized by passing through filters before being introduced. Depending upon the amount of oxygen required by organism, it may be supplied in the form of air containing about 21%. Then we can move on to antiforms. It is used to reduce the foam formation during the fermentation process. The foam is arised due to media proteins. It can be controlled by three methods: by using chemical agents, by mechanical foam breaker, and modification of the medium. Antifoams are surface active agents. That is, it can reduce the surface tension in the foam and destabilizing the protein films. Okay. An ideal antifoam agent should have some properties. That is, it should be active at low concentration, it should not be metabolized, it should not be toxic to the microorganism or humans. Then, inhibitors. Inhibitors are used to redirect metabolism towards the target product and reduce the formation of other metabolic product. Okay. Then last one, inducers. Majority of the enzymes are inducible. That is, substrate or substrate analogues are used as the inducers. Enzymes are produced in response to the presence of these compounds in the environment. Okay. So this is about media formulation. I think you got an idea about media formulation. Thank you.